welcome to our next Life Sciences revision lesson for grade 12, where we're going to be consolidating what we have learned about DNA and looking at some of the types of questions that could be asked. First of all, in the previous lesson, we learned that DNA carries the genetic code. In other words, DNA is like an instruction booklet for the cell, a recipe book for any substance the cell might ever need to make. And that also covers what cells need to do, because remember, cells make proteins according to the DNA, and enzymes are also proteins. So everything the cell does is also contained on the DNA. Where is this code found? And that is where the code is. The code is in the order of bases on the DNA. And those bases make up 3-letter words. Okay, and those are the base triplets or codons that we learned about in the last lesson. Now, what is DNA made of? You should be familiar with this. DNA is made up of smaller units called nucleotides. Each nucleotide consists of a sugar, a phosphate, and one of 4 different types of nitrogenous bases. Now, sometimes you will see a diagram that looks like this. Other times, you will actually just see a square with the base attached to it. Don't let that confuse you. When the base is attached to a square, the square is simply saying this is where the sugar and the phosphate are. It's not something different, it's just a simplified diagram to actually help you understand what is going on. So, DNA is made up of nucleotides. So, let's have a look and see how the nucleotides get put together. First of all, DNA has 4 bases: adenine, represented with an A, guanine, represented with a G, cytosine, represented with a C, and thymine, represented with a T. And if you have a look, you can see that because of their structure, because of their shape, C and G have to go together, and A and T have to go together. And we call those complementary bases because they fit into each other. So, adenine to thymine and guanine to cytosine. Now, each base is joined to a deoxyribose molecule, which would be here, and the deoxyribose molecules are joined together by the phosphates, which would form the sides of the ladder. So the sides of the ladder are formed by the deoxyribose and the phosphate, and the entire ladder twists into a double helix shape. Now, we've looked at DNA, but there is also RNA. And RNA is different to DNA because it is a single strand. Okay, here we can see it's a single strand, and instead of thymine, it has uracil. There are different types of DNA, of RNA, and we mentioned something about those in the last lesson. We have messenger RNA, which copies the code from the DNA. We have the RNA on the ribosomes, which put the amino acids together to form proteins. And then we have the transfer RNA, which is basically a label for each particular amino acid or ingredient, which the cell is going to put together into the protein. Now, it's very important that the amino acids get put together in the correct order so that the code can be correctly interpreted. So, in each of your cells, you have got DNA. Every cell needs to have all the DNA with all the instructions on how to do everything a cell will ever need. So, when a cell divides, it has to be very careful that it copies that DNA code exactly. And that is called the replication of DNA. And we want the copies to be absolutely perfect, because remember, this is the recipe that the cell is going to be reading when it's going to be making a protein that it needs. So it's got to be absolutely correct. So we're now looking at replication of DNA, and we're looking at it in this simple form. Okay, here is our piece of DNA, and the hydrogen bonds between the bases here are broken by an enzyme, and the two strands of DNA separate. Then, floating around in the nucleus, we always have lots of spare nucleotides, free nucleotides, and they would come along and they would join to their complementary bases. And that's how the DNA can make an exact copy, because the bases are complementary. Once the bases have lined up opposite their complementary partner, you can see what's happening here. These sugar and phosphate bonds are forming, and we will then have two separate strands of DNA. And if you look, exact copies. If the copy isn't exact, then we have something called a mutation, and that causes problems when the cell is actually trying to make a protein that it needs. Before we start looking at what you're going to expect in the examinations, I think let's take a break. Welcome back. I hope that break has refreshed your brain a little bit, because what we're now going to be looking at is what can you expect in the examinations? What types of questions can be asked? The first thing to remember is that even if you have difficulty with DNA as a section, there will always be some easy questions that you can answer. So focus on the easy questions, and don't be too upset if you get one of the difficult questions, because there will always be difficult questions. Let's have a look at the type of questions. Okay, first of all, you will always have straightforward questions testing, have you learnt your work? Do you know the names of all the parts? Do you know what adenine is, guanine, deoxyribose, etc.? And how do they fit together to make the DNA molecule? And you must be able to label the diagrams. Whether it's the simple diagram or the more complex diagram, you'll need to know that this is the deoxyribose, this is cytosine, things like that. Then there will also be questions testing your understanding. And one of the things you have to understand is, why does DNA have that particular structure? Why does it have complementary bases? And the important thing is that enables copying to happen, so that when a cell divides, the new cells have got all the correct instructions. So that's very important. You also need to know the process of replication, the steps of replication, and how one DNA molecule can replicate into two. Then the next one, and this is the one that we looked at in our last lesson, and that is how protein synthesis occurs. These are usually the most difficult questions, but if you bear in mind, you just think, okay, this is like making a cake. You will find it will be easier to actually follow through whatever questions they actually ask you. And we are going to do an example of one of those in a short while. Then you will always have your application questions. You will be given a different situation could be something you've never heard of, and you have to try and link it to what has been done in class. And this does require, for you to be able to do this, it does require you to have understanding, and it does require you to have learnt your work. Then there will also be something on practical skills, usually interpretation, for example, interpreting tables, interpreting graphs. Sometimes experiments are described to you, and then you're going to have to say, well, okay, if this is what's happening in the experiment, that is probably the result you will get. Or you could also be given an experiment that is described to you, and you have to analyze it in terms of the different variables, and then give things like a hypothesis and a conclusion. There's not a hang of a lot of the practical skills in this section on DNA. It's mainly tables, but every now and again you will get a graph. Then, last one, specific aim number 3. How does what we do in class, how does that relate to real life? And here you need to understand how we can use the technology from DNA in today's world. For example, genetic profiling or fingerprinting to do with forensics, finding the criminals, paternity testing, who's the father. Also very topical is the process of genetic engineering or genetic modification, in which scientists can actually alter the genetics or the genes of an organism. And there's a huge debate as to, should they really be doing this? Then, the effect of mutations. In other words, what can a mutation do? Can a mutation cause an organism to change the way it functions? And a very good example of this is the coronavirus, which keeps mutating and gives us a whole lot of different variants. So, let's now look at some questions. Okay, so we're going to start off with this one. This is a fairly simple one. It's a diagram showing a portion of a nucleic acid. First, you have to name the nucleic acid, and if you look at it, it's obviously DNA. And how do we know that? Because it is 2 chains joined together at the bases. Then you have to be able to identify what is portion V, and portion V points to this little square, and that is, of course, a nucleotide. Then base W, that's the one that's joined to C, so that's got to be G, or guanine. What is molecule Y? There's molecule Y, and that's got to be a P or a phosphate. And then bond Z, that's the bond between the 2 bases, and that is a hydrogen bond, which can easily be broken. Next, what is the natural shape of this molecule? Double helix. Now, where do we find this nucleic acid? Name 2 places in animal cells. Hold on a minute. It's found in the nucleus. Where else is it found? You should have heard of mitochondrial DNA because in the mitochondria there is a small little ring of DNA, so it's also found in the mitochondria. Then next, name the process during which this molecule makes a copy of itself. So the name of the process is replication, and then describing it would be those 5 steps of showing how the enzyme breaks the hydrogen bonds, the free nucleotides come and join their complementary bases, exactly what we were speaking about just before the break. Now, this is an easy question. Now we start getting to questions which are a little more difficult. You will always be given some type of source material, and in this case it is a diagram, and it tells us that the diagram is a process, is part of a process involved in the production of a protein. So this is obviously protein synthesis. Now, let's try and just work out, before you start answering the questions, you always have to work out, well, what is the picture actually telling me? Okay, so the picture is telling you that that is your messenger RNA carrying the code. This thing here is the ribosome. Because it's protein synthesis, that is your amino acid chain. Oops. And what would this be? This is bringing amino acids, so this is transfer RNA. Okay, once you've worked out what the diagram is actually telling you, you can then go and answer the questions. Okay, first of all, identify molecule Y, and molecule Y, we've already said that is the transfer RNA. Now, the group of nitrogenous bases labeled Z. Okay, it's just got numbers 1, 2, and 3 there. But what do we call those 3 bases? Okay, and that is the anticodon, which will match the codon which is on the messenger RNA. Next, what is this process called? And that is the translation process. You'll notice the first couple of questions are relatively straightforward, and we get to the more difficult ones a bit later. Okay, so if X is the next amino acid required after W, then identify bases 1, 2, and 3. And to do this, you have to understand that that transfer RNA molecule is going to be bringing number 1 to match with the next base, which is A. It's going to be bringing number 2 to match with the next base, which is C. And it's going to be bringing number 3 to match with the other C. Now, are they going to be identical to the A, the C, and the C? No, they are going to be complementary. So what is complementary to A? It's got to be U. What is complementary to C? It's got to be a G, and 3 also has to be a G. So that would be UGG. The next is the DNA base triplet that codes for X. Don't look at it and say, oh, easy peasy, it's ACC. This is the messenger RNA. The messenger RNA was made from the DNA. So what will the base triplet on the DNA be? It's going to be, first of all, what is opposite to adenine. Remember, this is DNA. Okay, so on the DNA there would be a T. What would there be next? To match with a C, there would be a G. And what would be next? Also a G. Now, this is where you often get caught. Just remember, the messenger RNA is complementary to the DNA, and it's also complementary to the transfer RNA. So, if we compare the transfer RNA and the DNA, have a look at it. On the transfer RNA, the anticodon is UGG, and the base triplet on the DNA is TGG. So your DNA matches your anticodon. Okay, so that's the thinking you have to do. Next, you will often be asked to show what you know about this process, and there you would be asked to talk about the whole process of translation, how amino acids are put together into proteins. Now, we can also have difficult questions. Okay, and this is one of the more difficult questions. It shows insulin made up of 2 long amino acid chains, A and B. A's got 21 amino acids, B's got 30 amino acids. And you've shown the different types of amino acids at the beginning of each of the chains. I don't know why that's not working. Okay, let's have a look at the questions. First of all, name the process, and that is obviously protein synthesis. Identify bond X, that is the bond between the amino acids, so that would be a peptide bond. Then, how many bases would code for the amino acids in chain A? Chain A has 21 amino acids, and each one of them is coded for by 3 bases, so it would be 63. Now we're getting to the difficult bits, because this table shows the messenger RNA codons and the amino acids, which you have to link. So let's have a look. Name the, first of all, an easy one. Name the nitrogenous base represented by G. G represents guanine, so that's quite straightforward. Now, this is getting difficult. Read the chains from left to right and give the codon for the 4th amino acid. 1, 2, 3, 4th amino acid. And that is GLN. So we have to look back at the amino acid table and find where is GLN. Okay, GLN is glutamine, and its codon would be CAG. Let's have a look at the next question, which is, what is the DNA base triplet that codes for the 1st amino acid in the chain? So we need to look back at our previous picture. The 1st amino acid in that chain A is GLY. So GLY, if we find it there it is, that's glycine, and its messenger RNA codon is GGA. So the DNA one that is complementary would be CCT. So in reality, working with these tables and the different amino acids isn't that difficult after all. You just need to follow step by step, and you'll get there. I hope that this has been of some assistance to you guys, and good luck in the exams.